Good morning, my name is Dr. Renee Shah-Mohammadloo. I'm a postdoctoral fellow working in the lab of Dr. John Fryxell, an eminent population ecologist here at the University of Guelph, which is a research-intensive learning university. I completed my program in aquatic toxicology, which seeks to understand the response of aquatic organisms to toxins that are naturally produced or human-made. In today's video, I'm going to speak about harmful algal blooms, which are dominated by cyanobacteria, commonly known as blue-green algae. This is a pressing issue facing our smallest to largest freshwater ecosystems in the world. Their expansion is due in part to changes in the global environment and increases in human activity, both of which are rendering freshwater ecosystems less resilient. With increasing frequency and intensity, harmful algal blooms threaten ecosystem services and freshwater biodiversity and have demonstrated the potential for adverse health effects in humans and wildlife. They've also been linked with massive fish kills in numerous fisheries around the world, with increasing records of poisoning cases connected to injuries from the toxins they produce, also known as cyanotoxins. In our lab, we are investigating the role of these cyanotoxins on the health of the water flea, also known as Daphnia magna. As part of Science Literacy Week, I've been asked to make a small video showing a day in the life of a scientist at the University of Guelph. With the theme this year being on climate, I'm going to show you a day in my life at the lab and the types of questions we explore. I hope you enjoy this video very much. So usually the first thing I do when I come into the lab is I take care of the Daphnia and green algae that are necessary for our experiments. Now, Daphnia magna are a very important species in freshwater ecosystems. The reason is because they are consumers of algae, but they are also consumed by higher trophic level species such as fish. When the population of Daphnia are impacted by stressors like cyanobacteria, that can impact our food web levels, and ultimately that can impact humans indirectly. So what I usually do is I start my day by culturing this green algae, which you see here, and this is a nice rich green color. And what you see over here in this tray are different genetic clones of Daphnia. Now what I mean by that is that there were 20 different clones of Daphnia that were collected at 20 different time points and spatially in different locations in a lake. The importance of that is because they're genetically distinct. And what I mean by that is that their genetic code looks different from one to the other. The reason why that's important for us in our research is because not all clones behave the same to stressors such as cyanobacteria and harmful algal blooms. And we're very curious to see if there would be a variation in genetic response. And the importance of that is because freshwater biodiversity is being threatened by stressors like harmful algal blooms. So by culturing these Daphnia, we can begin to answer whether, as climate is changing and these cyanobacteria are increasing in incidence, if they can impact freshwater biodiversity and if they can impact the diversity of organisms like Daphnia. So another important feature of my day is that I also culture cyanobacteria. This was provided by the University of Waterloo's Phytoculture Center. This is a toxic strain of a very popular species of cyanobacteria called Microcystis aeruginosa, and it's found in many places around the world in freshwater ecosystems. The reason that it is most intensively researched is because it produces a toxin called microcystins. And as you can see, cyanobacteria looks very different than green algae. It has this blue-green hue to it, and it's produced by a pigment called phycocyanins, like the color cyan. And that's what differentiates it from green algae, which has more of a green-yellow tint to it. So what I do part of my day is that I culture this cyanobacteria in the lab, and then I use it for large mesocosm experiments. And I'm going to walk you through one experiment that I'm running right now. And in that experiment, we're looking at the competition between blue-green algae and green algae, and then we put in those 20 different clones that are genetically distinct of Daphnia magna, and we want to see, first of all, whether cyanobacteria or green algae dominates in the face of increasing temperatures that's happening from climate change, and then whether that competition is impacting the diversity of clones of Daphnia that are present in the mesocosms. So this is the Aqualab facility, and you'll have to pardon the loud noise of the fans. It regulates the temperature in the room. So the purpose of this experiment, which I mentioned previously, is we're interested to see the competition between cyanobacteria and green algae. And when you add a diversity of clones of Daphnia magna, how the impacts the trajectory of these blooms. We're very curious to see whether the diversity of clones can, in a way, stop the spread and formation of these harmful algal blooms. And that's a really important question, whether genetic diversity matters in managing a stressor like harmful algal blooms all around the world. And as you can see from this tank here, there's a very scummy bloom forming, and we actually see variation from tank to tank. So I can see here that all the scum of the cyanobacteria and green algae. Now, if I peek in a little bit, it might be hard to see, but there are tens of thousands of Daphnia that are growing in these tanks right now. And it's very interesting because we collect these Daphnia and these water samples a few times a week, and we're very interested to see whether the numbers of Daphnia are increasing or decreasing. So in other words, to see whether the population is rising or falling as harmful algal blooms are rising and falling, and to establish whether there is a relationship between these two. Another important piece of this, and which is why I'm wearing this yellow suit, is because there is a toxin in here that's being produced, and for our own protection and safety, we wear these suits so it doesn't splash on our skin. So here's a closer shot. If you look at these little specks moving, these are the Daphnia magna that are all present at the top of the water column, and they are consuming the algae and cyanobacteria, as you can see here. These blooms can provide an abundance of algae, which contributes to their growth and reproduction. We're going to be collecting some water samples, which I'm going to bring these back into the lab and show you how we measure the levels of green algae and cyanobacteria present in each tank. So after I've collected all my samples from the Aqualab tanks, I then come back into the lab and I use a very important instrument here, which is called a phycoprobe. I'm going to plug in the phycoprobe and then talk to you a bit about what it does. So the phycoprobe is really important because it shoots out these fluorescent lights and it's collecting a whole wide range of wavelengths. And the importance of it is because it's trying to differentiate from, let's say, this sample here, how much of it is green algae and how much of it is cyanobacteria. So if I load it into the cuvette here, up to 25 milliliters, and then I put it into the instrument, it's going to tell me by taking over 100 images per second what the composition is. So let's have a look on the computer and see what we find. So looking at the data here, we see from that one cuvette that I loaded that there's a composition of green algae and blue-green algae. And it shows that there's about 80% more green algae than there is blue-green algae. And this is really important information when we think about how we're going to protect the Great Lakes, because an instrument like the phycoprobe can tell us by what percentage cyanobacteria, or blue-green algae, is dominating in our freshwater ecosystems. And the implications of this is really important because it gives us an indicator of how impacted species like Daphnia magna might be as harmful algal blooms are increasing in intensity during the summer season. Another really neat feature about the phycoprobe is that it measures and gives an approximation of how much of the cyanotoxins are present. And what's really neat by looking at the phycoprobe readings right now is there is a small percentage of cyanotoxins in the water. And we do know from research that cyanotoxins, and a very popular one is called microcystins, is known to impact all forms of life, like humans, fish, birds, and even the water flea Daphnia magna. And this is a really important feature of the FrequaProbe as well, because it can give us an estimation of how toxic a harmful algal bloom might be. Thank you for joining me in a day in the lab at the University of Guelph. I hope you benefited from learning about the impact of harmful algal blooms in freshwater ecosystems and the role that Daphnia magna could potentially play in curbing the severity of these blooms. I hope you enjoy your Science Literacy Week. Take care.